Today we're joined with Dr. Jean Hebert, neurogeneticist at Albert Einstein College of Medicine. The Hebert Lab its mission is to restore brain function with tissue engineering. And I just want to thank everyone for coming out today, and Jean for being on the show.
Yeah, thanks for having me on, Lowell.
Just as like a fun aside, because I was looking through your Twitter, I was looking through all the things that you talk about, and I've had people on who talk about neural engineering, brain-computer interfaces, and I always ask them, like, which one do you think will be more functionally restorative when it comes to TBI events? So if you had your bet, 10 years from now, 5 years from now, that type of thing, which one do you think would be more restorative? Brain-computer interface in terms of function, or a technology like yours that can go in there and either replace or rejuvenate damaged tissue?
Yeah, it depends for what. For spinal cord repair or bypassing the spinal cord, TBI will probably get us there faster because it can already bypass the spinal cord, right? So these machines can interpret intentions very quickly and then signal to the rest of the body to control motor movement. So we're already almost there. It's just that the devices are not yet portable, but groups are working on that. For brain damage itself, there really isn't much that brain-machine interfaces can do yet, because what they're good at is interpreting, your intentions. And if your brain is damaged, you don't even have the intentions anymore to be coded. So brain tissue replacement it seems more likely to be effective sooner than BMIs for that, because there are other reasons for that too that we can get into. But we have a blueprint for how the brain is normally built, which is during development. No one built our brains, right? They came from fetal tissue. Whereas for BMIs, they're sort of starting from scratch, and they really don't have a good way of mimicking brain function yet. So it's a less clear path, I think.
What made aging the thing that you wanted to dedicate your life to? And then what focused you to be a neurogeneticist to as the thing that you wanted to apply to combat aging?
Well, it was at a young age, the realization that we do age and fall apart and die. I never got okay with that. So, that was, I guess, a time when kids generally awaking to the world and what is the world about? What am I about? And then you realize, oh, in the end, you're just going to fall apart and die. And so that very early on was a trigger for me to start working on this. And, through high school, I started reading everything I could about genetics and how we can potentially control.
At any point in this conversation, if you find value in it, please subscribe. It is hugely beneficial, and it tells Google and everyone out there that this is content worth watching. Thank you for everyone thus far who has commented, liked, subscribed, and told their friends.
How we're made with genetics, and got my PhD in genetics eventually, because I really thought, the code of life, how could that not provide the answer? But then realized that for aging, it actually can't really provide the answer in any reasonable time scale, or certainly not for people that are already around today. And that's because a lot of what happens during aging is stochastic, complex damage to the components that make up our body: proteins, DNA, fats, sugars. And there is no genetically encoded pathways for reversing this damage. So, manipulating the genome or the epigenome might have some health benefits, but it's not going to get rid of aging or probably not even slow it down.
Then you parlayed that with how do you go from that to what you're doing now?
Yeah, so exactly. So I think a lot of the approaches that people are focused on currently for dealing with aging are going to have very little impact on aging itself, although they might increase health as we nevertheless age. I think the only way of getting rid of this stochastic complex damage that occurs to different classes of the molecules that make up our body is to replace tissue. And so that's the approach that we're taking is tissue replacement. And it makes sort of intuitive sense for the rest of the body, right? You can even conceive of replacing the whole body with, a headless body, for example. Mm-hmm. Take an old body, replace it with a young body, and then, your body's young again. Unfortunately, your brain is still old and still aging. And so does this apply to the brain? And I think it does if you do it progressively over time, because there's good evidence that you can do this without loss of function or self-identity. So that's what we're focused on, is to be able to progressively replace brain tissue, which in combination with replacement of the whole body or body parts can really make you young again.
With the grafts themselves, is it possible to get granular in terms of how the grafting works so that you'd be able to, let's say like you took like a snapshot of that part of the brain and you could layer it or set it up in such a way where it was more or less the same as what it was, or does it have to be kind of like fresh and then you don't do too much so it doesn't like override or destroy important parts of the brain?
Yeah, so the replacement process itself is not the most intuitive thing. So you don't look at how it's made right now and replace it exactly the same way. That would be next to impossible, because it's very hard to figure out what the connections are in a part of the brain without destroying it. So that would be very difficult to do. But there are two reasons why you don't have to do it that way, which wouldn't work anyway. One is that our brains, in particular, the parts of our brains that we use for higher cognitive functions, like what I'm using now to try to formulate, comprehensible sentences to you, and what you're using to try to decipher what I'm saying, the neocortex, that part of the brain is super plastic. So functions can move over time from, one part of that tissue to another part of that tissue without us even noticing. So that's been demonstrated across species that have neocortex and in humans of advanced age. So that means that you could potentially move functions from an old part of the brain to newer parts of the brain without you even noticing. The other part of the equation here that makes this worth considering and worth pursuing as an approach is that, again, we didn't build our neocortex, right? It developed from very simple structures in the fetus, where these cells are essentially pre-programmed to give fully mature functional tissue. So, and people, many different labs, including ours, have transplanted these cells and demonstrated that they don't seem to care that they're in the adult environment or even a damaged adult environment. They go about their business of trying to give rise to new functional tissue. Those cells don't succeed because we're not putting them in there the right way, and those are the things that we're figuring out, is how to do that, how to put them in there so that they develop and differentiate and integrate normally with the adult brain.
How long, I imagine there's like a number of steps between where you are now and something that someone could be using, even for compassionate reasons. I mean, you're still in the mice models. What would be like a rough timeline before something could be more what's left of the technology to develop, so that you can translate it out into a startup or license the IP for someone to develop into something that would be directly applicable to people?
Yeah, I think we can start, developing IP immediately, because in order to reverse engineer this early tissue, which can give rise to functional new tissue in the adult brain, we're making a lot of sort of discoveries of how things work, which are useful and have value intellectual property wise. Not that that's what we're after here. We're really after a way of replacing brain tissue with new brain tissue. But there certainly is a role for industry to play in this, to accelerate the progress that we're making. Currently, our academic lab, is making rather slow progress on this front, and having industry or private partners, whether philanthropic or with commercial interests, come in and accelerate this will be greatly beneficial to us. But you were asking about the timeline too, like how long. I mean, we're trying to set up a company now for the purpose of accelerating this. We have some committed funders already, but we haven't kicked off yet. But even with that, even if we got all the money in the world, I don't think we could have a tissue ready before 5 years to try people. And that's really like the fastest imaginable possible time before going into humans. It could, of course, take a lot longer, 10 to 15 years. Or if things don't accelerate and, we don't get support and sponsorship for this, it could take, 50 years or
What would your role be at the startup? Would you stay in your lab and be like a George Church, like helping people out, or would you be like the CSO or something?
Yeah, no, I'm committed to this project, whether this project is in the lab or if the project transitioned to industry or some type of institute or whatever the mechanism for moving it forward. I will be 100% involved with this project. So I'm not anchored in academia, and I'm not necessarily driven to be just an industry. I'm really guided by what will make this project move fastest forward. And yeah, I would be sort of hopefully involved, if not heading the scientific development of this replacement tissue.
How do you think about the adjustment in timeline when you have an academic lab versus a startup or something like that? I know many people who've made that transition, and the hardest thing is getting people to not have the academic mindset in terms of developing something, even in terms of how they structure teams and whatnot. So I'm curious, as you're thinking of the company, I imagine there's a part of you thinking like, how's the production going to look like? What that change look like?
Yeah, it's really the benefit, again, is acceleration, right? That comes primarily from funding. In an academic lab, it's very flat over time. You publish papers, you renew your grants, you publish more papers, you renew your grants. So funding and progress occurs at a very flat rate, I guess. Whereas in industry, you meet your milestones, and then the money gets bigger and bigger exponentially. So you can really start to get things done. And so the idea is that with more people working on this over time, a lot of the problems that we face in terms of, should we do it this way, or should we try it this way or this way? Instead of trying one, then the other, then the third, we could do them all in parallel, get an answer much quicker. So meet our milestones much quicker, and make progress in developing, again, this replacement tissue that we look forward to doing.
That makes sense. The parallelism. Is there a startup or industry lab that you've gone to, I think they say great artists steal, so like steal their methodology to running a startup type lab versus an academic type lab?
Yeah, no, I have to admit, I don't have experience in industry, even though we're going in that direction to accelerate the project. So, I'm certainly not of a corporate nature in terms of running things. I very much like to have open discussions and debates about the approaches we're taking, and I look to hire people that are of like mind, who are want to discuss, think about, and then try, that aren't afraid to try and fail, because there's a lot of that in the R&D that we're undertaking. So that's certainly the culture I would foster. Beyond that, there's also a sense of urgency. We can't take too long to
Hmm.
get this done.
Yeah. And then, do you know the amount you're trying to raise? Like, there are a lot of venture capitalists that listen in, so maybe someone out there's like, oh, that's in my pocketbook.
Yeah, sure. Anybody listening who's interested, that would be great. So we're before we start hiring, we want to have somewhere in the order of 2.2 million uh, in the bank, and we're like just over 75% there now.
Mhm.
But then we'll keep the seed round. So this is seed round funding. We're going to keep that open for a while to increase it, maybe 100% before closing the seed round. And then, of course, after that, it's all dependent on milestones and yeah.
Yeah, that makes sense. Well, anyone out there, reach out to Jean. The In terms of your technology, I was curious what you think about it as a compare and contrast for Michael Levin's work, where he sees the hardware of things. He sees like there's a hardware and then there's a software level. I don't know if you're familiar with Michael Levin. I think he's
Yeah,
kind of
yeah,
near. Okay,
yeah, yeah.
so what do you see the difference of approaches? Where his is software level, where he just puts the right material in that area and then he gives it the right chemical bath essentially, and then the cells will regrow and do it that way, versus your method, which seems more you graft it in and then I guess it's not too dissimilar actually. Now I'm thinking about it, but yeah, how do you see that?
There is in fact quite a bit of overlap there because
Yeah.
the goal is to generate new tissue, and however you do that, it's a good way of replacing all the pre-existing damage.
Mm.
And I think because of the complexity and stochastic nature of that damage, the only way of really rejuvenating the tissue or reversing aging in that tissue. So yeah, so there is overlap. I think for the brain, a big problem is the lack of stem cells. I say that and people go, oh, no, there's stem cells in the brain. Yeah, but they're restricted to very small areas. Much is made of them, but they have very limited potential, very well encoded fates. So it's not like they can replace neocortical tissue, for example. They're very restricted to another part of the brain called the dentate gyrus, which is a small part of the hippocampus. And it's actually still a little controversial whether the stem cells in humans are there, but there's enough evidence to suggest they might be. And so there are, unlike Michael's work, there are no cells that we can that are there that we can coax to generate new tissue. So instead of that, we put them in ourselves because we know how to make them from human-induced pluripotent stem cells. And again, it's not just one cell type, right? It's whole tissue regeneration, which again is what Michael is doing. So, kudos to him for taking that approach.
Mm-hmm. Yeah, I wonder if at some point his technology might interface with your technology as you replace what's there for his stuff to work in. That might be some interesting synergy. But so neocortical precursor cell types. When I was reading your work, and I tried reading as much as your research as I could with being a layman. How do you decide what cell types to include and what not to include? Because you don't want like too many monkeys in the kitchen.
Right. Yeah. So we're 100% guided by the way that it's done normally. So we have access to human fetal neocortical tissue. And so we do, high resolution analysis of the cell types that are there early on. And there are, at least a half dozen different cell types that are required for normal development of the tissue. Even though, like I said, many labs have taken the strict neocortical precursor cells for the principal neurons, transplanted them in adults, and they do remarkably well. They don't seem to care that they're in adults. They project long distances and connect with the brain, but they don't make functional tissue, so they're not really useful to the host. So it's only by really recapitulating normal neocortical development that we can hope to achieve functional tissue. And so we just base our choice of cell types on the ones that are there normally. We try to embed them in an environment, so an extracellular environment that has the structural and chemical components that are normally there as well. So we do like proteomics analyses, and we're going to start some glycomic analyses as well to try to get that right. And then we have to ensure that the interaction of those different cell types is occurring early on normally, because they are key to development later on of that tissue. So I mean, those are the things we're really focused on getting right. But we're not reinventing anything new. We're just sort of reverse engineering what we know already works. So that makes our task, in a way, a lot easier. I mean, it's still a big project.
Yeah.
But we don't have to invent anything new.
Yeah. Is there a cell type in particular that is like the workhorse cell? And then I guess the joke would be, why is it not glial cells? But is there one that like does more work than the others?
I mean, everybody thinks as the precursors of the principal neurons as the most important. And in a way, they are. It's like we need every part of our body to be alive, right? If we don't have kidneys, we die. If we don't have lungs, we die. If we don't have... But if we were going to pick one that we think is the most important, we'd probably pick our brain, right?
Yeah.
Because that's who we are. So the same thing at a cell level in the neocortex. All the cell types are essential for the development and function of the tissue. But, we kind of like the principal neurons because they're the ones that convey the bulk of information processing, even though they can't do it normally without the other types of neurons that modulate their activity. And that's what most people transplant when they transplant a single cell type in the neocortex. They transplant the precursor to these principal neurons. So, yeah, that's... I don't know if I answered your question on that.
No, it did. I you never know when you're interrupting or not, so I just let people talk. But what prevents scarring? So I worked at a brain-computer interface startup for a little bit, and the introduction of electrodes, you had to find, I don't want to get sued for IP stuff, but you had to be concerned about these things. So, but you're making lesions and you introduce new tissue to it, and you even you add some media to get the blood out, so before you introduce it. But what's preventing the scarring from happening, like any buildup or anything like that?
Scarring is a concern, and something we definitely are always keeping an eye on. But we've managed to minimize scarring just by using strong anti-inflammatories at the site of the lesion. Once a lesion is made, we basically soak it in these steroidal anti-inflammatories, which seems to help quite a bit. And then shortly thereafter, we'll introduce our starter tissue. We seem to detect very little scar tissue between what we put in and the host tissue, and lots of vascular connections and neuronal connections between the border of the two. So we're cautiously optimistic that we can move forward with that. But it is a concern that if there is any scar tissue, it can inhibit normal neuronal connections with the host.
Do you have to keep the anti-inflammatory? I imagine that, like an organ transplant, I think to some extent you have to suppress the immune system so your body doesn't fight it off. For the transplanted tissue, do you have to do a similar mechanism? Like, do you have to continue administering that over the course of the life of the organism, or is it just for that period where it's healing?
Yeah, well, the anti-inflammatories is just for the period when it's healing. But the immune suppressants, those are necessary for longer periods of time when you haven't matched the cells to the donor, when they're not autologous. and especially when they come from another species, then you need immune suppression at fairly vigorous levels for forever. But that's not what we plan on doing. It's just preclinically, we'll test our human structures, our human starter tissues in mice. And so those mice have to be heavily immune suppressed. When we do it in humans, if we do non-patient matched, like not derived, if the cells and the components in the tissue are not derived from patient cells, then we will also need to do immune suppression, as they do currently for cell transplant treatments for Parkinson's, and there's others as well that are in progress in clinical trials. So there's a pathway for doing this. There's protocols for doing this. In the brain is a little different than the rest of the body. You can escape the immune system more easily. So a lot of these approaches in humans where they don't use patient-derived cells, they taper off the immune suppressants quite a bit over time. Mm-hmm. And the cells still seem to survive. But ideally, we want to go to patient-derived. So the iPS cells derived from the patient that we're putting back the brain tissue in. That way, we don't need to use immune suppressants. We have to be very, very careful, though, that there are not any undifferentiated stem cells in there, because they give rise to teratomas. But again, these things have been done in humans already, and there are protocols for, really minimizing the risk that you do have any undifferentiated cells left.
Are mice the best model outside of experimenting in humans to do this type of research? I'm thinking of at UW-Madison, they use rhesus monkeys and other type of primates when they're doing brain studies.
Yeah. So, I mean, best only in the sense that there are the lowest, sentient mammals, right? But not so good for what we eventually want to do, which is proof of concept that the tissue we've put in, by virtue of its electrophysiological activity and integration with the rest of the brain, is now encoding a useful function to the host. That is much harder to do, especially if we're trying human tissue at that point, in the mouse than in like an old world monkey like rhesus macaque or another type of old world monkey. So those things would give us more definitive answers if we don't use rodents.
Mice have the added benefit of they're faster lived. Is that like a component that makes it easier for when you're doing academic or translate it into a startup type scenario? Just the fact that you can do lesions and see the full life in a shorter period of time to see if there's any developmental issues?
Not for what we're doing. For a lot of cases, yes.
Yeah.
Absolutely. Especially if you want to look at effects on maximal lifespan in a mammal, well, they have one of the shortest lifespans, so they're ideal for that. But for what we're doing, it's not really an advantage. Their short life actually limits their usefulness to us, because the human brain, takes from the early stages of fetal development to when it starts to be conscious and functional, let's say, sometime after birth. You're talking about at least a year or two, and then it continues to develop beyond that. And so mice, don't even live that long. Mm-hmm. So again, in terms of testing the human tissue that we're making, mice are limited both in the size of the neocortex, but also in their lifespan. So again, the ultimate test may have to come in primates. whether that be non-human primates or the human primates.
Yeah. So people, I'm wondering the implementation of a lot of this that we've been talking about. And one person, this will be AI related. One person, a listener asked, with the explosion in AI technology recently, does Jean have any? Is that impacting the future of brain regeneration? And is there anything AI related that you're working on now in the lab that you intend to incorporate, or that you already are incorporating it, or that you're going to be doing in your startup as well? And this person's name is Phyllis 075, which is a great name.
Yeah, I don't think I know this person, but yeah, thank you for the question. Yeah, we're not using anything AI related at the moment in the lab. And in terms of discovery or engineering the tissue, I can't conceive of a way that we can train an AI to be useful on that, because the information is difficult to find. Where AI would be generally useful for people in the field of medicine and health sciences would be to have something like a chat GPT that has access to PubMed, right? So all scientific publications. So right now, they don't have access, so we still have to gather and go searching for information ourselves, which is somewhat time consuming. I mean, that's one sort of silly way that AI could be helpful in the near term. Other ways have more to do with automation. So a lot of what we do is fairly labor intensive, but could be encoded in an AI that was trained to recognize growing cells in culture, because we do a lot of that, right? We need to generate a lot of these brain cells in culture from iPS cells or stem cells. So automation in the culture procedure, training the AI to recognize, what are good cells, what are bad cells, and when to feed them, when to passage them. All these things could potentially be automated, and would be wonderful to have, and would take, that would take like smart AI combined with smart robotics to do that. And I think there are some groups working on that, but we haven't implemented yet, and hopefully it will come soon. The only other aspect that great to see some progress with some AI are some smart surgical robots. But again, right now there's still, people are developing these as well, but they're still sort of controlled by a surgeon. But eventually maybe they can be more independent.
How would you use the surgical robots?
Again, just for the implanting our starter tissue into lesion brains, for example, that would require a robot can potentially see much better than us, can control hand motions much better than we can, or movement of the tools much better than we can. So the risk of hitting a blood vessel, for example, can be diminished.
Okay.
Yeah.
That makes sense. And then a part of what you're doing is genetically engineering microglia that disperse through the adult neurocortex to bolster the neocortical function. So two-part question there. Why microglia? And then is the engineering technique to engineer them?
Yeah. So that's a completely different project. So, there's really two projects in the lab. The big one is the tissue replacement one. That's what we're mainly focused on. But there is a residual project that is very interesting as well, using microglia as a way of delivering either cells or biologics like protein therapeutics, like antibodies, for example, to the brain. And the reason that's sort of interesting is because right now there is no way of widely delivering payloads to the brain because of the blood-brain barrier. So if you systemically inject these protein-based therapeutics, They have very short half-lives, but they also don't cross the blood-brain barrier for the most part. So you can't deliver them to the brain. So this project is to use microglia as sort of little delivery mechanisms for the brain, because if you what we can do, and others are doing this as well, is you can ablate the endogenous microglia transiently. They're important. You don't want to kill them off permanently, but if they're not there for a short period of time, it seems to be fine. And then instead of having them repopulate from the few that are left, which they do, they're very driven to repopulate the entire central nervous system with new microglia when you do deplete them. Instead of letting them do that, you put in your engineered microglia and they repopulate instead. And that way, if the engineered microglia are engineered in the sense that they're secreting like a good neurotrophic factor or anti-inflammatory factor or whatever you want for whatever disease you want, then those microglia are there sort of producing this good factor over time. It's sort of a one-time treatment as well. And they distribute like throughout broad areas of the CNS, which is needed for something like Alzheimer's or aging, which affects all parts of the brain. So if you want to deliver neurotrophic factors, they could be a good way of doing that.
So for either approach, do you have so Alzheimer's is one of the neurodegenerative disorders that you'd have, damage to the brain. What are some of like the key implications of the illnesses that you'd be able to help with, or damage you'd be able to help with?
So for the microglia, the advantage there is widespread delivery.
Mm-hmm.
Right. We're looking at more broadly degenerative conditions like Alzheimer's and aging. For the tissue replacement, it's initially we're focused on local damage like stroke or trauma, since it's the only way to actually reverse aging, what we want to do is implement that the replacement of new tissue progressively for large areas of the neocortex and eventually the rest of the brain as well. And so that is a step removed from immediate applications. We first have to show that we can make a tissue that is functional. Once we can do that, then we can consider wider spread damage like what occurs in aging.
And we have a longtime listener question coming in here with neurodegeneration. Town Grizzletown, which is a great name. What are your thoughts on how amyloid-beta aggregates relate to neurodegeneration?
Oh yeah, they want me to get into that controversy.
Yeah, I was going to ask it a different way, but this one sounded more technical.
Yeah, well, again, the evidence isn't great that they're a central driving factor of the disease. And so I think we need to look, those people who are interested specifically in Alzheimer's need to look beyond that and be open to other approaches to combating this disease.
Do you have a pet theory? Either that you think people are going in the right direction, or that you think like maybe they should spend more time on this?
Yeah, I'm talking to a lot of people working on Alzheimer's, and they each have their different theory. I'm not an Alzheimer's specialist. Some think it's a vascular disease, or it's just inflammatory. It's the microglia. It's one thing or another, or it's amyloid, or it's tau, or it's something else that happens earlier. Whatever's happening, it's happening in an old brain where everything is compounded by the fact that things are falling apart. So my approach is, let's just replace the tissue progressively over time. So now you have a young brain and you won't have Alzheimer's, you won't have aging, and these other problems.
Would you be able to replace the whole brain, or only just the neocortex?
Yeah, so the goal would be to replace the whole brain. Every part of the brain is essential for normal information processing, the way we think of it as normal at the moment. And every part of the brain ages and falls apart. The neocortex might be the most studied part of the brain in terms of how it develops. But for other parts of the brain, we have a fairly good understanding as well of all the early cell types that are there, and that form that part of the brain. So the idea would be, when we want to reverse brain aging, and we know we can do it for the neocortex, is to also develop it for the other parts of the brain that would make sense to replace at the same time as certain areas of the neocortex. For example, the visual neocortex, right? It goes through the visual thalamus, which is a different part of the brain than the neocortex. And so having, and you can do it unilaterally, so having the cells that normally connect together and grow axons during development be there doing that in the adult brain at the same time would probably be the best way to get normal function again. So you might want to do like sensory modules of different parts of the brain at the same time. But this is something that needs to be, worked out and tested. The associative parts of the human neocortex, those seem to be so plastic that I don't think that those could probably be implemented at any time or coincidentally with these more primary functions of the neocortex, like visual processing, auditory processing, sensory processing.
Yeah. So my follow-up question to this is also related to a question that someone else is asking. So I'm going to ask theirs, and then jump, like, twist it to where I want to go. But Paraphyllis 075 is coming back in. How invasive would this type of procedure be? Are we talking removing parts of the skull and applying the treatment every time? And if so, will the idea be to create less invasive options over time? Could it be done via needle? Yeah, I was thinking as well, like, if you have to, like, I just imagine like a pie, like, you take like 5% every time, but then that sounds pretty horrible as a way to mentally think about it. So yeah, how do you see how would you stage the change? And then I guess answer Paraphyllis 075.
Yeah, 5%, 15%. We're thinking more 15% based on the rate of plasticity of the human neocortex in people of advanced age. But still, that means, like, maybe quite 6 to 10 brain surgeries. But yeah, there, it is brain surgery. So you would make a craniotomy, access the brain, do the procedures, and then put the skull back and let it heal. This sounds undesirable, but I don't know that there's a way of replacing tissue without doing that. And again, when you talk to neurosurgeons, which we've done because we're looking ahead to the clinic, this is something they do every day at work. It's just a job for them. And so it's not like this is something that doesn't occur all the time in hospitals across the world, every day. It does, right? It's something we hope as individuals we don't have to do. But if it means, reversing the age of your brain without loss of function or self-identity, I certainly would be open to that. And I'm guessing others might be as well.
I've read of people, I've read of people that they go through one of the jugulars, or I think it's like through your leg or your armpit, or I don't think they do the neck one. I don't know why. But anyways, they go up that way and then they'll cut apart a tumor with little scissors, and so and then like slowly suck it out. So I was wondering, if you use microrobots, could you just cut out the section that's bad and then suck it out, and then like squirt out the new layer of brain, and that way you wouldn't need to crack open someone's skull and do. I feel it would be less invasive.
Yeah, I mean, that if that technology looks like it could be used, that would be, of course, much better. And it's not impossible, as you describe it. It's probably easier, but still a good step in the right direction to be able to just like cut up a tumor and suck it out than to rebuild a starter tissue on site. But maybe not impossible.
Yeah.
Yeah. So, yeah, anybody wants to work on that in the meantime, please.
Yeah, you can buy up the IP when it's ready, and it'll have a home to be used. We talked about timeline. We talked about staging, how long it'll take to slowly increment it over. So I always wonder when it comes to the mice populations, and if it was in the paper, and I glassed over it, my apologies, but when you replace it, when you look at the baseline function, and then after the how long does it take to get back to where they were in terms of functionality?
Yeah, so the other issue, so it would happen, I think, within 2 months, right? So we did do some physiological tests of our graphs to show that they respond to visual cues, and they acquire that responsiveness over the course of 2 months, which sort of follows normal development in pups of the same part of the brain. So really our graphs, again, are trying to do exactly what normal developing tissue does. So it takes about 2 months in mice. For human grafts, it takes a lot longer. So, they really don't start coming online until 6 months, and then they're still, developing and differentiating for at least another 6. So it takes a lot longer. And that's what you would expect the time frame to start to see that tissue being used by the host. The difference, though, between mice and primates is the level of compensation in mice is remarkable. Mice seem to be less dependent on their neocortex than we primates seem to be dependent on it. In mice, you can bilaterally lesion the entire motor cortex, and within a week they'll be up and running, behaving normally, scratching, eating, drinking. In humans, you do that, you're paralyzed. You don't. There's very little recovery of function over time, if any. And so it makes it a little more challenging to test certain things in mice when they're able to compensate even without new tissue so quickly. So we're a bit limited in that sense as well, which is another reason why eventually we need to do this in primates of one species or another to ensure that it works.
Is there a limit on when you can apply this technology? It doesn't sound like let's say I had an injury like 5 years ago, and I have not 100% yet. It seems like you just cut around. You just cut a little bit more, and then you'd add more, I guess. It doesn't seem like there'd be a limiting time factor in terms of when an injury happened or when you could be applying it to a person.
Yeah, I mean, if you have an injury and you have some tissue there that is non-functional, like scar tissue or something, and either removing it or displacing it and rebuilding a tissue underneath it, if that's possible, then you could probably do it at any time. The more progressive loss of functional tissue that occurs, for example, with aging, that requires, as well as the progressive addition of tissue in, maybe 15% at a time over the course of, probably a couple of decades, would also require removing the old tissue, right? And so I mentioned that the plasticity that exists in the neocortex, where functions can move from one part of the neural substrate to another over time. And that has been shown in humans for things like language, our personality. But it happens when there are the documented cases, when there's a slow destruction of the original tissue from a pinpoint out. And that happens when there's benign gliomas, so brain tumors that are benign and operable. So they'll destroy the original tissue over time, slowly enough, a couple of years, from an initial pinpoint out. And if you're still using the function that was encoded in that location during that time, it just gets re-encoded seamlessly elsewhere. So this has been observed in humans in their 50s, 60s, 70s, where they then realize they have a brain tumor because they had a headache and went to the hospital, or had a seizure because of the pressure and went to the hospital, and had it removed. So now there's a big hole like in their language center, but they never lost the ability to speak. And when the surgeons check to see where the language is now encoded, it's a different part of the neocortex. So this gives us, the precedence for being able to remove tissue at a slow rate as well as add tissue. So if we silence, and we have the technology to do it, or at least to test it, if we can silence, 15% of the neocortex over time, then we can remove that tissue, the old tissue, making room for new young tissue at the same time.
Yeah, now I'm following you. Is this the best way to do that, to achieve that functionality? Is there like a way to use pluripotent stem cells or something else to be injected that would, cannibalize the damaged tissue and then regenerate itself from there? Can you steelman any other approach besides the one you're using and banking your life on, I guess, is the question.
In terms of replacing the tissue, both the adding and removing aspects of it?
I think on a higher level, just restoring function. Because if I wouldn't want you to like argue against a different approach to removing and replacing the tissue. Because that's
that was sort of the idea, right, with the microglia as well, is we have means of getting them in there and having them spread, and we could potentially convert them to new neurons, right, through cell reprogramming. So that's been done ex vivo. It has, for microglia, it hasn't been done in vivo. It's been done for other brain cell types in vivo. So it's conceivable that you could add new neurons sort of dispersed throughout the neocortex. But it gets very complicated because, neurons come in many different types, and they're organized in a very in a way that matters to their function. So in the neocortex, they're organized in layers, and that's important for the information processing. And so just adding new neurons, what type, are they going to be there in the right ratios compared to the other neurons? Are they going to be in the right place? It quickly gets complicated and much harder than just putting in new tissue. New starter tissue that can develop the proper composition and organization of cells on its own. This is an approach that we thought about and actually invested a lot in early on, until we realized that it just doesn't make sense to try to overcome all these problems when the tissue, if we put it in right the first time, we'll know how to fix all this by itself. So, yeah.
Yeah. All right. Transition. Halstead19, who has been a long-time listener, has a question. They're wondering your viewpoint on the role of extracellular matrix, ECM, and the damage that needs to be addressed. While most cells appear to have the capabilities to self-replicate, the ECM does not. Would he consider an approach that focuses only on replacing the ECM, leaving the rest of the work to stem cell therapies, senolytics, etc.?
Yeah, I think that's a very good question. The extracellular matrix, I think, is where much of the age-related damage occurs over time. And again, this is the stochastic forms of damage that have been well documented for long-lived extracellular proteins. It happens in many different ways. There's all sorts of forms of covalent modifications, breaks, aggregations, non-covalent aggregations. So it gets real ugly over time. And a lot of that is extracellular. And we know this to some extent because we've done what I call heterochronic transplants. So taking young brain cells, putting them in an old brain, and guess what? They behave like old brain cells. Or taking old brain cells and putting them in a young brain, and then they behave like young brain cells. So their extracellular environment is very important to how they're going to function. And I think you could go a long way if you could reverse the extracellular damage. You could go a long way in reversing or at least extending the life of a brain by doing that. The difficulty, though, again, is that this damage is complex. There's no genetically encoded machinery for dealing with it, so you have to now invent new machinery that doesn't exist that can specifically target like covalent bonds that are damaged and not touch ones that are normal, which is very difficult. Although the person asking the question, I think, may have possibly been alluding to more wide scale, just like, let's just degrade the ECM and then have cells produce a new one. Again, conceptually that would make sense, but implementing it without, again, losing who you are and might be very difficult to do. Again, I don't see a way of doing that. There's no way of getting rid of this cross-linked damaged protein. So you'd have to invent some new technology there. Is it impossible? No, but again, I don't see an easy way forward to doing that.
Well, Halstead, maybe there's an opportunity for you to get a PhD and solve this very problem. Related to aging, you never know. They could be a researcher right now, and they're just looking for more, jurisprudence and other ideas. But related to aging, cosmic existentialist. That is a fun name. Are you aware of the thermodynamic theory on aging? If the theory turns out to be true, would current aging and neurological degeneration therefore be futile? And they gave me a link, so I can read it if you don't know it.
Yeah, please do.
Okay, good. So the thermodynamic theory of aging explains changes of the functions of states of cells and tissues during aging. The rate of aging depending on the genetic factors, the nature of habitat, nutrition, and external influences. These rates can be different. Aging organisms basically just describes it longer. I imagine it's like the thermodynamic thing. It's like one of the 3 laws of thermodynamics. I don't know physics, so I don't know how to ask this question better.
Yeah, there are multiple theories of aging. I'm not a big fan of theories of aging. I think we know enough about what happens with aging. Again, this damage to the extracellular matrix is a big part of it, but also DNA damage, and damage to other classes of macromolecules, which are understudied, probably also occurs. But we know there's enough damage there that is a driving force. Again, because by itself can cause all the other hallmarks of aging. So this damage is something that we need to address if we're going to beat aging. And so getting back to the question, maybe you could reread the question about trying to think of how this thermodynamic theory might...
I don't understand it. Cosmic, please just comment, and maybe I'll just post it to them, because it just seems like it's the natural degradation, the aging of organisms, functional systems, and tissues as their diseases lead decrease of adaptability of organism, and eventually its death. So it's just like normal death. I'm sorry, Cosmic. I don't understand this question either.
I mean, it's hard to disagree with that statement.
Yeah.
Right.
It just feels like normal aging. So then,
Yeah.
Cosmic, I hope you don't make fun of me for doing a bad job answering your question. So, and then just add a comment, and I'll send it to Jean. So, Grizzletown, what new town Grizzletown for the record? We talked about startups and being spun out of your research lab. Are there other ones besides the one you mentioned previously? And does it have a name? Can you say these things, or are you like under wraps? Like, so what other startups have spun out of your research from your lab is the main question from Grizzletown. And then I peppered you with 2 other ones.
Yeah, I mean, we don't even have a webpage yet, but the tissue engineering company is called BE Therapeutics.
You could still, is there time to change it to Lowell Therapeutics? Like, no reason. I just feel like it'd be great. No, okay, I'll stop.
And then there is a microglia spinoff as and the person taking the lead there is someone else, Robert Cargill, and so he's co-founded with me a company called Glionics.
You mentioned on your website. I'm sorry. Go ahead.
No, that's it. Yeah.
Okay. You mentioned on your website that you're always looking for people to join your lab, and you have 2 startups that are spinning out with your technology. What are there core people that are core skill sets, core experience type people that you're looking for that's hard to find? Like, there's some people it's like, I don't know, maybe easier to find, but like who are like the rare gazelles out? I don't know what the analogy I would use there, but what are the rare types of people that you're looking for that you can't find?
Yeah. So there's a couple of people we're looking for right now for BE, the tissue engineering company. It stands for brain engineering. One is a tissue engineer, someone who has more experience with developing biogels that have the properties that we want. So we can do a lot on our own, but in terms of optimizing things, we could use a lot of expertise there. That's the main thing there. We can also use somebody who has some experience with surgeries. Again, we do all of those ourselves, and we collaborate with surgeons as well. But to have someone full-time on site doing some of that would be helpful.
This is all in the New York area, right? You're not like moving this to a lower cost of living place.
Yeah, for now we're in the New York area. Oh.
Yeah, I'm going to suggest the Midwest because Madison has a great place and it's cheap. But I had a commenter saying that it's annoying that I suggest that so much. I don't know if you can cut the cost by a third. Why not? You're building something pretty hard. Do you have any plans to move out of New York, or is it just like that's your home? So there's a fixed cost there in terms of where you're willing to move it.
It's sort of the same as home being academia or industry. I don't care. It's where the project is. So if the project can better be done elsewhere, I'll move. No problem.
Sweet. I'll get you a lab space out here. What books would you recommend people check out or read? Either related to your work, unrelated to work. You do have a book that I'm going to link into the show notes, but even the things that you enjoy in your free time. Is it mystery, whatever? Sherlock Holmes? Yeah, what books?
I haven't read a book for a little while.
Oh, wow.
Gosh. Yeah, I'll have to come back to you on that.
Well, we can recommend yours. What's the name of your book?
Oh, it's called Replacing Aging.
There you go. Then what advice would you give to people who are out there? The typical age of the podcast seems to be about 25 to 35. Though there's a big section of like plus 55-year-olds that are listening in. What advice would you give to people who want to get more involved?
Yeah, I mean, first thing is to, I think, educate yourselves as much as possible on what's being tried now to address aging, and be very critical and seek out opposing views to try and make up your own opinion, because there are prevalent views out there, and some people just drink the Kool-Aid and are cheerleaders for a certain approach when really, in my opinion, that approach has zero chance, and yet it's a very prevalent view that this is worth trying. So be critical, get educated, and then depending on your skill set, there could be ways that you could help disseminate information yourself, or if you have means, help fund certain projects you think are worthwhile, or if you have any a science background, then there's probably room for you to directly work in some of these projects because they are very multidisciplinary. I mean, Lowell, you brought up a couple of areas that we're not doing, but that could very well complement what we're doing, in addition to the stem cell work, the very wet lab type of stuff that we do. That's what I would suggest, I guess. But yeah, do get involved. We need more people involved. There are networks as well for, Nathan Cheng has this network with
Lifespan Bio, right?
Yeah. And so contacting them to see where you might fit in best to help the overall fight against aging, that would be a good starting place as well.
Is there a place for people who are self-taught? I guess that the coders for machine learning up solution for you would be one of them, but there's a lot of people that don't have PhDs necessarily. So then, is there a place for someone like that?
Yeah, if they have the skills, the degree is kind of irrelevant, I think. I have a student in my lab who is completely self-taught and does a lot of the computational analysis, so, along with a little bit of coding for the high resolution analysis we do of fetal tissue to try to reverse engineer it again, to make sure, for example, that the cells we're generating in the lab match the normal cells that are there in a human fetus. This is called single cell sequencing and the analysis that goes with it. And he's completely self-taught, so he doesn't need, it's good that he did that. He's also trying to get a degree at the same time. Mm-hmm. But you certainly don't need a degree to be able to be very useful and helpful.
Yeah, unless you want to be a surgeon, you have to get a degree. We're not letting anyone go up the street and have that role. Yeah. So enough concentrate is probably going to be our last question of the day. Enough concentrate is related to surgery, so that's a on-point joke. Can you predict any non-surgical methods of tissue replacement? I asked you this question earlier. For example, intranasal delivery that might have lower friction to use in terms of... I'm thinking about the ways that surgical procedures make medical treatments much more infrequent and require further disease progression to justify. I can't exactly go to the brain doc for a tune-up by taking a long lunch. That's true.
Yeah. No, this is not a, something you do over your lunchtime. Not yet, anyway. There are other delivery, routes for cells like intranasal or CSF, so like through the circulating cerebrospinal fluid, as opposed to intracranially directly by making a hole or a craniotomy. But again, for what we're doing, those right now don't seem possible. The closest non-intracranial approach might be the one you mentioned, Lowell, where somehow you can smartly go up a vein or a blood vessel and then be able to, reconstruct what we want. The issue again is that the starter tissue that's needed to get normal mature tissue, the organization of that tissue has to be right, and you can't do that just by injecting cells in the circulation, in the cerebrospinal fluid, or intranasally. And that's actually been the big roadblock to making progress in repairing the neocortex. People have been putting cells in there for a century, and repair has not occurred yet. And that's because no one has put in something that looks remotely like normal tissue, because it doesn't have the structure. So we really need to get the structure of that tissue to be normal for the function to be normal.
How do you? So if there's a graft sitting in front of us, there's a brain, there's a graft in it. So there's a little I don't know inverse piece of the brain missing. How do you layer in the cells right now to be in there? You don't just like wad it up beforehand and then inject it. So I meant, it sounds like you like layer it in a specific order.
Yeah, so there's some layering that can be done in vivo, but again, there's some preparation of the cells that can be done ex vivo, so before we do the graft. There are certain key aspects of brain development in general, but particularly for the neocortex, that we need to recapitulate. So certain interactions between the cells are known, across mammalian species to guide this layered structure that we need to achieve to get normal function. And some of that we can prepare a little bit before grafting and then do more once it's grafted as well.
I was thinking of when they build a house and they're putting the foam up, there's some that you roll out, but there's some that's just in a hose that you spray it down and then you layer it outwards. So I was thinking if you're coming from some type of ventricle, you go in there and just like layer the one that's closest to the skull, and then like a cake. Which I hope that's not as simple as that, but maybe that would work. And if so, I trademark that, and you have to pay me. Would that work?
All in the details, though. Yeah.
Yeah, true. I don't trademark that. Don't worry. So for aging, you mentioned that there's it is hard to tell who are the people that are probably better to be watching and some of the people who aren't. And I'm not gonna have you be mean to anyone. So who would be some of the people that you recommend people just be following their work? Because there are some that I found people say the nice things on the website, they'll say nice things in public, but if you start looking at the details, like you said, the devil's in the details, and there's definitely some devils in those details. So who are the people that actually work out to be angels versus devils when you look into the details?
Yeah, so I like Alexander Fedintsev. He's certainly has the right perspective as to what needs to be reversed in aging if we're going to be aging. So he understands the complexity of the damage, and that is what we need to address. He's more focused on the ECM. Some argue that DNA damage is the driving force of aging. I don't think that's the case. I think it's a contributing force. The ECM, there's a stronger case to be made that it's more of it kills us sooner than DNA damage does. But still, I think they all need to be addressed. But at least Alexander, has the right appreciation for what is aging and what it is that we need to be fighting. And then this may go back to this thermodynamic model of aging, right? Things are falling apart at that level, and so the approaches he's taking, I think, are more to target the ECM specifically, which again, I find that difficult to imagine how he might successfully do that. But I still think he's worth following what they're doing.
Right.
Then there are other people that are not considered as anti-aging researchers or life extension researchers that I think are really important to follow. And those are the ones that are building replacement parts for the body. Mm-hmm. A lot of them are building biological replacements. Again, if you replace a old body with a young body, we know the young body will last longer. So we already have a proof of concept. And so, there are people working on replacing whole body at once, but there are also others that are working on replacing body parts like organs and different combinations of organs by growing them in the lab so that they're devoid of any age-related damage, and then transplanting those. One of the biggest groups working on that is Anthony Atala's group at Wake Forest. He's got a big operation there, and they've had some success, but I think they're definitely somebody to keep an eye on. People are also working on synthetic replacements. Nod Seston at Yale is a very interesting, completely synthetic replacement for the body called Brain X. I don't know if you've heard of them.
No, I haven't. I'm just wondering what would be the
You
replacement.
may have heard the headlines. I think they took like pig brains an hour after the pigs died, and were able to keep the brains alive and metabolically active without a body. So a completely synthetic, replacement, a circulatory system that filters the blood, oxygenates the blood, removes the CO2. It's pretty wild, but if you think it's okay to be a cyborg in the future, have a biological brain but be in a completely cyborg body, I'm always excited to see what they're coming up with as well.
Yeah, my preference is to try to slow or hinder any technology that ends up with me being a brain in a jar. So, but I also see the value of the technology, so I won't be mean to them. But that does sound interesting.
Yeah. No, I mean, brain in a jar, it doesn't seem worth it. But if that brain is well matched with a brain-machine interface, that where you're getting input as well as being able to control, your artificial limbs or whatever, then it's not so bad, right? You actually wouldn't know the difference. Your brain wouldn't know the difference whether the visual perceptions you're getting are coming from an artificial retina or a real retina. Your brain wouldn't know the difference. And then when you move an arm and you see it move, but again, I don't know that you would know a difference if it's done well. We're not there yet. We're talking future now.
How do you know that you're not a brain in a jar, and that what you know was stunted so that you don't think it's possible, so that you don't question it like in the Matrix?
Yeah, we don't.
You don't? Okay. You seem pretty comfortable with it, so all right. I guess it's all the same.
Yeah.
All right. Well, I want to thank everybody for coming out today, especially you, Jean, for sharing your knowledge and tolerating my jokes. Thanks for coming on the show today.
Yeah, thanks for having me, Lowell.
At any point in this conversation, if you find value in it, please subscribe. It is hugely beneficial, and it tells Google and everyone out there that this is content worth watching. Thank you for everyone thus far who has commented, liked, subscribed, and told their friends.